Good morning, good afternoon, good evening, everyone. Thank you very much for joining us for another episode of Emerge. My name is Jan Stojcev and I'm from the Resin Bio team. And today I have the pleasure of hosting Jason Dirks. Before I introduce him and we go into the main session, just the usual housekeeping announcements. We are recording the session. We're going to make the presentation available on our YouTube channel, so check out Resin Bio on YouTube, and you can find this as well as previous recordings. Please post your questions in the Q&A section. We'll have plenty of time to get to this and answer these. Also, we like to encourage a live discussion on this platform. If you want to ask your questions live, please use the raise hand icon. It's in the middle or at the bottom of your screen. And I will activate your microphone after Jason is finished with this presentation. So Jason is currently completing his PhD in the Slavov lab in the Northeastern University of Boston. He's, I think, doing his 5th year now, so almost done with the PhD. And he's mainly been focusing on improving data acquisition workflows for mass spectrometry, improving their workflows, improving their throughput as well as sensitivity. Today he's going to be talking about some of this work. He's been focusing on multiplexing data independent acquisition workflows and how this has benefits in terms of improving the proteome coverage as well as reducing missing values. So Jason, first of all, good morning to you. Thank you very much for taking the time to join us on this platform. And I really look forward to your presentation.
Yeah, thank you so much for inviting me to talk. And thank you for that introduction. So hopefully you can start to see my slides. Is that correct?
Yep, it's coming through.
Yeah, okay, great. Yeah, okay, yeah. So thank you for that introduction, Stoyan. And thank you everybody for tuning in to my talk. Today I'm going to be talking about plexDIA, which is a method that we've been developing here in the Slavov lab and in collaboration with Professor Demichev. And what this seeks to do is increase the throughput of proteomic analysis. And we do this by multiplexing data independent acquisition. So what are some examples of why somebody might want to increase the throughput of proteomic analysis? And some examples of that might be that they may want to increase the statistical power of the analyses that they're doing. And so to do this, one might want to run many replicates. Another example might be maybe you have large scale experiments. Maybe you have bulk samples with a bunch of different conditions, and otherwise it might be a little bit cumbersome to run all these samples back to back. Likewise, maybe you have single cells, you want to have a data set with thousands of single cells. And so these are all different possibilities of why you might want to do high throughput proteomic analysis. And this can be achieved in a few different ways, one of which is on the LC side, where you have like really fast gradients, you have a high flow rate. And in this way, you can just run higher throughput analyses because the data acquisition, the method length is actually going to be shortened. Some downsides to this, though, is that as you increase the flow rate, you tend to reduce your sensitivity. On the other hand, you can also increase the throughput on the mass spec data acquisition side by using shotgun DDA. So you can run samples in parallel by TMT multiplexing, and 18-plex is commercially available. Or you could run it with a DI method, which is going to increase your proteomic coverage. So I'm going to talk about these methods a little bit more in detail. So yeah, the goal is to quantify as many proteins as you can, as accurately and quickly and repeatably as possible. And one proven method is to do shotgun DDA, where you can do TMT multiplexing. In this case, you have these samples that you are running in parallel. So you have these precursors that are eluting in this MS1 scan that are basically a superposition of each other. And you're going to sequentially send each one of these precursors for fragmentation and then an MS2 scan. And you use these backbones, which are going to be a superposition of each other across these samples to share this identification. And then you go and extract signal for quantitation at these expected reporter ion M over Z's. And so this has a lot of advantages. It has the advantage that you have an 18-plex, that you can run 18 samples in parallel. The other advantage is that because you are running samples in parallel and you're sharing in this identification, extracting quantitation, you can have really high data completeness within a set. Potentially, what some of the downsides, though, is that the coverage is, limited by time. So you can only, so you're limited basically on time based on the number of MS2 scans that you can take. So if you were to do a shotgun DDA method, basically you could only quantify one precursor per MS2 scan. So if you could only run maybe 20,000 MS2 scans, the most precursors that you could quantify and identify is about 20,000. The other thing is that this method is using reporter ions for quantitation. And this can result in co-isolation. For example, if there's a precursor that falls into this window, the reporter ions that you get are going to be a superposition of these precursors. So this could result in potentially slightly lower accuracy sometimes. The other potential downside of this is that you have low data completeness across sets. And this is because the sampling is data dependent and it's going to be slightly stochastic. You're not always going to analyze the same precursors for everyone. An alternative to this is a method that is independent of the data. So you take big chunks of this MS1 space, and you send everything for MS2 fragmentation in each one of these windows. You can identify all the precursors in this chimeric spectra using some DIA software such as Diane. It's going to deconvolve these spectra, and you can identify these, and then you can quantify them based on these MS2 fragments or based on this MS1 precursor intensity. And this has a lot of advantages. For example, you're less limited by time because with each MS2 scan, you can potentially identify hundreds or thousands of different precursors in a single MS2 scan. The other thing is that the quantitation is not dependent on reporter ion, so you're not going to have, you'll still potentially have interferences, but it's not going to be resulting from co-isolation. And the other thing is because this is a method that is independent of the data, the results that you get, the data completeness is going to be a little bit more repeatable and reproducible. The downside, though, is that it's most widely used in a label-free manner, and so you can really only run a one plex at a time. But there was some really nice work that was done by the Gigi lab a few years ago where they showed that using an 11-plex TMT, running samples with 11-plex TMT and doing offline fractionation, you can get really deep proteomic coverage, quantifying about 8,000 human samples quantifying about 8,000 human proteins in about 3 hours of acquisition time per sample. Similarly, that's actually about what you can get with running a label-free DIA method. And this is, so one does this, the shotgun DDA approach does this by multiplexing samples in parallel, whereas the DIA method does this by analyzing peptides in parallel. So if you would think about it in a way of, if your goal is to get deep proteomic coverage, you could think of the throughput of label-free DIA and shotgun DDA with multiplexing as being approximately the same. So we wanted to use both of these. And so we were thinking of using plexDIA, which, we're going to do data independent acquisition, but we're going to multiplex with isotopologous chemical labels so that we can run, not only acquire peptides in parallel, but acquire samples in parallel. And this is not a novel idea. This is something that people have tried previously, but yet it still has not been well benchmarked against a label-free DIA method. So it's still sort of unclear whether you could actually increase the throughput multiplicatively by running samples in parallel. And so some of the advantages of doing so is that you get all the advantages that you would get with a DIA method, and pretty much all the advantages that you would get with a shotgun DDA method that does TMT multiplexing. You get deep proteomic coverage, quantitation that is not subject to reporter ion co-isolation. You get repeatability of a data independent method, and you get multiplexing and high data completeness within a set because you're running samples in parallel. Some of the potential downsides, though, would be that you have increased spectral complexity because you are running 3 samples in parallel. This could manifest in lower accuracy because it is more complex, and could result in lower sensitivity, so lower coverage. So these are all sort of theoretical, pros and cons. And so we wanted to see how well this could actually work. And so we collaborated with Professor Demichev, who is the lead author and inventor of DIAN, which is a really popular DIA search software. And we set the goal of optimizing DIAN to sort of interpret this plexDIA data. So I'm going to run through a really quick example. Let's assume that we have 3 samples which we labeled with isotopologous labels, and we're interested just in looking at this one peptide sequence. So this peptide sequence is going to co-elute in a single run, and you're going to have these different isotopes eluting at the same time. Because the label that we use is a mean reactive, it's going to label N-termini and it's going to label lysines. For this particular peptide, though, it doesn't have any lysines in it. So we can only use B ions. So if you have fragmentation of this ion, you can only use the B ions for quantitation, because these are the ones that are going to have a mass specific difference. These other Y ions, though, are going to be shared. You're not going to be able to separate this in MS2 space. That doesn't mean that you can't use them for identification. In fact, you can use them for identification, just cannot use these Y ions for quantitation. So let's go through this, and let's assume that we searched it kind of in the same way that you would a label-free DIA experiment. And we find that this label N has the lowest Q value. So it has the highest confidence, has the one that has FDR less than 1%. These other 2 co-eluting other samples have higher FDR, but there's still valuable signal in these channels. So what we would like to do next is re-extract the signal from those less confident ones. And also, let's pretend that there is a decoy channel. So let's pretend that if the MTRAC label has a mass of 140 Daltons, and that's the lightest one, and the next one has a mass of 144, and the other has a mass of 148. Let's say that there's this theoretical label that has a mass of 152, and let's go out and extract signal, if there is any, for that decoy. And then let's train a classifier to separate out what is true from what is false. And so we can do this and get a channel Q value based on taking correlations between the most confident eluting precursor, the one that had FDR less than 1%, and our actual real targets and our decoy. So let's see if we can, train a classifier to do this for every single run. And so this is a good idea in theory, but how well did it actually work? And so we set up this experiment where I have, E. coli and yeast. They're all labeled with the lightest isotopologous label. And then we just have yeast labeled with a slightly heavier label. So what we should expect is that the yeast peptides are going to co-elute. They're only going to be separated in this M/Z dimension, but they're going to be co-eluting. And E. coli is only going to be in this delta 0 label, and it's going to be by itself. But DIAN is going to try to integrate signal for it in D4. And so now the question is, how well can this channel Q value actually filter out whatever spurious signal this might be integrating? And it actually works quite well. So if we look at the MS1 precursor intensities, we find that the empirical FDR matches the channel Q value that we set. And this is true for an MS1 level and for the MS2 level. So to summarize this, basically we can translate identifications. We can share in this confidence of the identification because these samples are co-eluting, and then we can go out and extract signal where we expect it to be in the isotopologous labels in the other samples. And this can operate in 2 modes. You can share the identification and extract signal like I was just discussing. And this is analogous to SILAC. It's also analogous to TMT. Or you can have really controlled translated FDR using the channel Q value filtering, where you will. So this could maybe be applicable to things where you use like immunoprecipitation, or where you have a mixed species sample where you have expectation that maybe some of these proteins are not actually in the sample. So you can do either of those. So in terms of quantitative accuracy, this was the other concern, that because we are running samples in parallel, there's going to be more complexity, and the complexity might manifest in lower accuracy. And when we first started doing this analysis, it did seem like that was the case. But then we actually found ways around this. We found that, we can have really strong accuracy with at mostly the apex of the elution peak, where the signal is greatest and the impact of potential interferences is the lowest. And so we came up with this translated quantity algorithm, which basically it takes a ratio of the fragments that are in common with the most confident label to the other fragments that maybe were not as confident. And you take ratios of these fragments. For example, this light blue one is maybe 20% more abundant in this confident one than it is in this other one. And this dark blue purple one is in a 1-to-1 ratio. And when we form a consensus based on these apex ratios, that now we're basically saying that this label is 10% more abundant than this less confident one. And then we can basically integrate the signal for the more confident one, like summing the fragments over all the scans. And for example, if this one came out to have a quantity of 1,000, we would then multiply this by the scalar that we have calculated here, or rather divide it. And then we would say that this one has a translated quantity of 909. And in theory, this is good, but how well did it actually work in the data that we have? And I'm going to explain where I got this benchmarking from. It's going to be in the benchmarking section, but I thought I would show you the data right now that it does in fact improve the accuracy that you get at an MS2 level. So having MS2 quantities as you normally would versus MS2 translated is more accurate. So to summarize this, we found that quantifying precursors and precursors at the apex gives the highest accuracy. We found that we can use a translated quantity to improve MS2 level quantitation. Okay, so this is all good in theory, but how well does it actually work? And so we sought to answer this question. So we made 3 samples, and each of which has a mixed amount of predefined species. So we have one sample that has a certain amount of E. coli, yeast, and a human cell lysate, monocytic cell line, so U937s. And we made 3 of these, each with different spiked in amounts of E. coli and a serum of ACA. And we can use this for quantitative benchmarking. We can also use it to compare the number of identifications that we get. So we kept these label-free, and we also aliquoted some so that we can label them with isotopologous labels. And then we combine those labels to form just a single set that we can run one at a time. And we did this with 2 different data acquisition schemes. One that is MS1 optimized. It's going to take really high resolving power MS1s infrequently. And the other is a MS2 optimized method that is going to take many MS2 scans and it's more so optimized to give deep coverage. And so this is basically what this is going to look like. For label-free DI, you have to run each sample by itself. But with the plexDIA sample, the idea is that you can then run everything at the same time. So it takes 1/3 of the amount of time to acquire the same samples. So if we look at the coverage, plexDIA can identify actually about 2.5 times as many precursors and proteins per unit time than label-free DIA can. It's not exactly 3-fold more, but it's about 15% or so less in terms of absolute numbers. But we can actually make up for this because we are running samples in parallel. We have really high data completeness because we can share in this identification and go and extract signal like you would in a SciLac experiment. And this is what I'm showing here. We're showing the data completeness for plexDIA is actually almost 100%, and we can quantify more proteins than you could in common across the samples than you could with label-free DIA, and you can do so in a third of the time. And again, you can do this because you're sharing in the identification, and you can translate and extract signal where you'd expect it to be. And here is another panel where I'm going more into depth about what's going on here with this data completeness. And so we ran 3 replicates of these samples. And what I'm showing here is a heat map showing the protein Jacquard index, which is just kind of another term for data completeness. So if you have a value of 100, so 100% data completeness, it just means that all the proteins that you identified in one sample, you also identified in the other sample. We find that, yeah, when you are running samples in parallel, you can have very high data completeness. On the other hand, though, with label-free DIA, you start to have less data completeness, especially as the proteomic composition gets more and more different. And I'll touch on that in just a second. But we also ran shotgun DDA method. And we found, as expected, the shotgun DDA methods, they tend to be a little stochastic. You're not always going to sample the same precursors with each replicate. And so for that reason, the data completeness for a shotgun DDA method is lower than it is for a DIA method. And this is something that you would expect. Now, what's really interesting, though, is if we look at the missing data between these pairs, so I've just kind of flipped it. Instead of looking at data completeness, we're looking at data missingness. And if we look at the ones that were run with the same samples, with plexDIA and label-free DIA, they actually have really similar amounts of missing data. And the reason this is because, for example, we're looking at sample A in the first replicate to sample A in the second replicate. The reason they're so similar is because they're subject to the same variability, and that variability is chromatography. So these other samples, so if we compare different samples though, label-free DIA, which I mentioned before, has less overlap in samples that are more proteomically distinct. And if I refresh your memory, samples B and C are actually very different. They have 6-fold difference in the amount of E. coli, 6-fold difference in the amount of yeast. And they're actually comparing 2 different human cell lines, monocyte and a T-cell cell line. So as we would expect, these 2 different sample types, which are very proteomically distinct, have a lot of data missingness, actually the most in comparison to the other pairwise samples. Likewise, A to C was the next most amount of missingness, and then A and B. But with plexDIA, because you can run some of these samples in parallel, so the within run data missingness is actually very low. So we've kind of shown that the coverage, albeit might be a little bit lower on the absolute terms compared to label-free DIA, we can make up for it with data completeness within a set. So next we wanted to look at quantitative accuracy. How good is the quantitative accuracy compared to label-free DIA? Because we're doing this 3 times faster, are we suffering from maybe worse accuracy in terms of quantitation? What I'm showing here also is just showing the number of protein ratios that we can get. And this kind of recapitulates the results that we were saying beforehand that, with similar proteomic composition, plexDIA and label-free DIA can do about comparable in terms of coverage, but plexDIA, when it comes to very different proteomic compositions, such as comparing samples B and C, you get a lot more coverage with, or significantly more coverage with plexDIA than you would with label-free DIA. And the quantitative accuracy, which we applied, we did some LFQ bench experiments and plotted it. Basically, we know what the ratio should be. And so we just plot what our actual values are. And we find that we have very high accuracy with plexDIA compared to label-free DIA on average. And so what I was showing you just now was plexDIA within a set. And so the question then is, how does it actually compare? Say you wanted to run a large data set and you have many runs, how well does the quantitation for sample A in the first replicate, for example, compared to sample B in the second replicate? So this is a cross-replicate quantitation. And quantitation actually looks just about as good. This is what I'm showing here is the absolute value of the error for both of these methods, or for in-set quantitation versus out-of-set quantitation. And here's showing the sort of LFQ bench experiments for plexDIA and label-free DIA as well. And if we look at the MS1 and the MS2 quantitation, both are actually fairly accurate comparably. One thing that is interesting that we find is that these, but maybe not surprising, is that lysine peptides are slightly better quantified than arginine peptides at MS2. And the reason this is because you can use B and Y ions because they're sample specific for these lysine containing peptides. Whereas these arginine containing peptides, you could only use the B ions because they're the ions that are going to have this mass tag. These other ones are going to be superposition of each other, and you can't use it for quantitation. So because you can have more fragments with lysine peptides, the accuracy of the quantitation could be a little bit better, but they are still reasonably comparable. So the next thing that we did was we checked the quantitative repeatability of plexDIA in comparison to label-free DIA. So we ran the same samples in the triplicate, like I was just mentioning before, and we looked to see how repeatable is the quantitation that we get for plexDIA, how well is the repeatability of the quantitation that we get for label-free DIA. And you see that the median is actually about the same between plexDIA and label-free DIA. So this is a good thing to see. However, there are labeling biases. And these are sort of an artifact of any sort of method that has labeling, is that you introduce, artifacts. This is kind of ubiquitous. But what we found here, so what I'm showing here is we have sample A and it's labeled with a delta 0 channel. And then we also have sample A and it's labeled with a delta 4, and then sample A labeled with delta 8. So we find that the CV, while still being reasonable, is a little bit higher across these labels. But this is something that you could potentially correct for with a large enough data set that you can use batch correcting algorithms to reduce the effect of this repeatability issue. Okay, and so next we wanted to, we've shown that plexDIA and label-free DIA can give accurate quantitation, but do they agree? And so we used these samples that we have. One is monocyte cell line, U937s, and the other is the Jurkat cell line, T cell. And so we took ratios of the proteins for these different samples, and we did this for label-free DIA. So for example, if we had a protein that was 5-fold enriched in U937s compared to Jurkats, then we would expect to see it to be 5-fold enriched for plexDIA as well. And this is something that we see that for all proteins, the Spearman correlation is about 0.8. And then for the proteins that we found to be differentially abundant in label-free DIA, the quantitation is actually recapitulated quite well with plexDIA. Presumably, these are proteins that you would really care about the quantitative accuracy for, and it's nice to see that we can recapitulate this with plexDIA. The other thing that we wanted to show is that we wanted to assess whether the statistical power of plexDIA and label-free DIA is comparable, and it does seem like it actually is. So what I'm showing here is we have true positives on this y-axis. So we know that Cerevisiae and E. coli are spiked in at different amounts. So we should see that they are in fact differentially abundant. And we also know that the human samples contain the same cell type, and so they should have no differential abundance. And so if they are, then you take a hit to the empirical FDR. And so what we find is that plexDIA and label-free DIA tend to have about the same statistical power, and it's almost indistinguishable below 2%. And you can do this about 3-fold faster with plexDIA. So to summarize, what we found in the benchmarking is plexDIA, you're going to get, maybe a little bit lower coverage per sample. This is also something that you find with pretty much any labeling approach, is that you tend to identify things to a lesser degree than you would if it was label-free. And some of this might be side reactions. But we can make up for this by running samples in parallel because we can have higher data completeness. The accuracy seems to be comparable. We have comparable repeatability in terms of coefficients of variation. And there's strong agreement between plexDIA and label-free DIA quantitation. We also have comparable statistical power with plexDIA. And so I think we can say that, we can increase the throughput of plexDIA seemingly multiplicatively. And in this case, we've used a 3-plex using MTrack, which is what is commercially available currently. So this is great. We've shown all these things that, plexDIA can potentially increase the throughput of proteomic analysis. But then we also wanted to apply it to a biological system to see if we can actually do some real biology with this. And so we decided to analyze the cell division cycle of U937 monocytes with plexDIA. And so we have actively dividing U937 cells, and we stain them with DNA, and then we fact sort them. And the ones that have like 1x DNA are going to be the G1 phase. The ones that are actively replicating their DNA are going to have somewhere between 1x and 2x DNA content, so they'll fall somewhere along here. And the ones that have twice as much DNA content are going to be in the G2M phase. So we sort these three populations. So we have a G1 phase, an S phase, and a G2M phase. We label them with the isotopic labels. We combine those 3 samples into a single set, and then we label, and then we run them with the 2 data acquisition schemes, which I mentioned previously. One that is MS1 optimized, it's going to use MS1 quantitation, and one that is more optimized for coverage, that's going to use MS2 quantitation. And then we acquire the data, and then we wanted to, take a view of what this data is like from 30,000 feet. What is the systems biology processes that are being upregulated or enriched across these different cell cycle phases? And we find some ones that are really canonical to what you'd expect for a cell division cycle analysis. We find that the MCM complex, which has helicase activity and is required for DNA replication, is enriched in the S phase. And we find that the mitotic sister chromatid segregation among cytokinesis after mitosis and spindle associated processes are enriched in the G2M phase. We also find that the V1 method and the V2 method, two very different data acquisition schemes, one also using MS1 quantitation, one also using MS2 quantitation, have very good agreement between these different processes and the enrichment that they find with these different processes. What's also interesting to find and keep in mind is that we're doing this in the context of immune cells. So these are monocytes. So it would maybe make some sense that maybe some of these immunological pro-inflammatory processes might be enriched in certain cell cycle phases rather than others. And maybe that is the case. We do find RAGE receptor binding processes to be enriched, as well as positive chemotaxis and peroxidase activity and hydrogen peroxide catabolic processes to be enriched in the G1 phase. So aside from this systems perspective, we wanted to get a little bit more detailed, and let's actually look at what are the proteins that are statistically differentially abundant across these cell cycle phases using these different data acquisition schemes. And we do find some really canonical ones as well. So we find that UBE2S, which is a protein that ubiquitinates anaphase inhibitors, is enriched in the G2M phase. So this would potentially allow cells to proceed through the M phase and eventually divide. We also find that polar-like kinase, which is involved in several different mitotic processes, is enriched in G2M phase. We also find a lot of proteins that seem to be involved in chromosome segregation and seem to be spindle interacting are enriched in G2M phase as well as proteins involved in DNA replication and repair seem to be enriched in S phase. What is also really interesting to find is that we have a lot of metabolic processes that seem to be enriched in different cell cycle phases, suggesting that there's some coordination of metabolism with the cell cycle. And so what I'm showing here is glycolysis and the pentose phosphate pathway. And what we find is that the G1 phase seems to be enriched for the non-oxidative phase of the pentose phosphate pathway. This is shown with TKT, transaldolase, and then this enrichment actually stops right before it gets to this irreversible reaction that leads into the TCA cycle. And then we found that the TCA cycle, if we go back to the systems perspective, looking at the protein set enrichment analysis, TCA cycle was actually enriched in the S phase. So this does, make some sense. What we also found, which was, kind of interesting, is that the glucose transport is enriched in G2M phase, and we find that this protein, hexokinase 2, which is a protein that phosphorylates glucose, is also enriched in G2 phase, suggesting that glucose is potentially being transported in at a greater degree at the G2M phase, and that you have this protein that is now phosphorylating this glucose and then trapping it inside the cell. We also had some proteins that, comparatively are less characterized. And we wanted to, check to see, we found that they are differentially abundant, but we wanted to have a little bit more confidence to this measurement. And so we extracted the ion chromatograms for these precursors, for a precursor for these proteins. So we're showing this right now for at the MS1 level and the MS2 level, so fragments at the MS2 level. And what we're finding is that they are co-eluting together. So they're basically apexing synchronously because we are running this in parallel, and that we find that they are being enriched in the G1 phase relative to the S phase for both these proteins. So with this, we can have a little bit more confidence that despite these being relatively poorly characterized, if we look at the raw extracted ion chromatograms, it seems to recapitulate exactly what we'd expect. And it shows that they are, in fact, eluting synchronously. So with this, we showed that plexDIA can be applied to study, real biological systems, and it can do so at a multiplicative throughput. So we were running 3 samples at a time. The G1 phase, the S phase, and the G2M phase were combined together. And we found some canonical things, exactly what you'd expect with different cell cycle phases, and we found some things that are maybe slightly less expected. So what we've shown here is that this can work quite well at a bulk level, and then we wanted to analyze what can this actually do if we go to more sensitive analysis. So let's analyze single cells. And so I had the help from a fellow grad student, Andrew Leduc, who's a graduate student in the lab as well. And we used this method that he's been making, NPOP, nanoproteomic sample preparation. And with his help, we sorted some cells into a really tiny droplet of DMSO. So 8 nanoliters of DMSO, we sorted, 1 cell at a time. This would lyse the cells. We add trypsin to digest the proteins. We label it. And then we add hydroxylamine to quench the reaction. And then we pool these cells together, each of which is labeled respectively into a single set. And then, of course, we alternate the labels to avoid, potentially any systematic biases. And then we ended up with about 60 sets. And we acquired 10 of these sets on a timsTOF STP with the help of Matthew Willits, who is a proteomics scientist at Brooker. And this is showing the duty cycle that we used for this analysis. The duty cycle length was actually very quick. It's about 2/3 of a second here. And the data that we got, we identified about 3.5 precursors per cell, and then about 1,000 proteins per cell as well. And we did this on a 15-minute active gradient. So it took about 5 minutes of active gradient per cell, and we're getting about 1,000 proteins per single cell. And we ran about 50 sets on our first generation Q Exactive, an instrument that was commercialized about 10 years ago. And we used a really simple duty cycle, just a single MS1 scan, and then 4 MS2 scans with variable length, M/Z lengths. And the duty cycle length for this was about 1.5 seconds, and we used 70K resolving power for this. And we identified about 1,400 precursors per single cell and about 500 proteins per single cell as well. We did this on a 30-minute active gradient, so 10 minutes per single cell. So if we look at the data completeness that we get, we actually have almost near perfect data completeness with the timsTOF SCP. And some of this would be attributable to the instrument, having higher sensitivity. Some of this, a large part of this though is the fact that we are running these different cell types in parallel. And so we can share in this identification and extract signal where appropriately for these other isotopologous channels. Similarly, the Q Exactive actually has good data completeness as well, about 80% data completeness within a run for plexDIA sets. So, this is good. We've shown that we can get good coverage, relatively quickly. How good is the accuracy? And to answer this question, we made samples that are, pretty similar to our single cell sets, but we had 100 cells. Instead of just a single cell of each type, we have 100 cells of each type. And we ran those, and we wanted to quantify, or we wanted to see what is the agreement of our average real single cells to this bulk experiment. And so we should find that the quantitation should agree. And actually it does quite well. For the timsTOF SCP, we have Spearman correlation about 0.8. And the Q Exactive, we have a Spearman correlation of 0.84. And one thing I should say is actually that these bulk samples that we got were acquired on the Q Exactive. And they actually came from a batch of cells that were prepared, weeks beforehand, weeks before these actual single cells that we ran on the timsTOF SCP were made. The reason I say this is because not only are there probably some technical biases that are potentially, lowering this correlation because we ran it on different instruments, but also there might be some small biological differences between these cells. So the reason I'm saying this is that I expect the correlation for this to actually be a little bit higher than 0.79 with a fair comparison. So the next thing that we did is we identified which are some of the proteins that are differentially abundant in this analysis that we did. And I picked some cells that are I picked some proteins that kind of fell along this range, and some of these proteins are, one of these proteins was HMGA1, which should be enriched in PDEX about maybe 5-fold or so. And this is something that we see in the extracted ion chromatogram for our single cells, that we do see this to be true at the MS1 level. We see it to be true at the MS2 level. And we also see that they are apexing synchronously, which is exactly what you would expect when you run these samples in parallel. We also find that tubulin beta is enriched in U937 cells relative to PDEX, which is exactly what we would expect from this analysis here. And keratin 7, which I should say could be, in other analyses could be interpreted as a contaminant, but there is a lot of literature that says that keratin 7 is, maybe 50-fold enriched in PDAC cells relative to white blood cells, which U937 cells are a white blood cell. And the fact that we get this result in our bulk analyses, and it's also recapitulated in our average single cells, suggests that this actually is a real biological signal. And what I'm showing here, the reason I'm listing the C-scores is something really interesting is happening. We are able to leverage the fact that these samples are being run in parallel, and we get a really high C-score in the PDAC channel. So C-score is kind of inversely proportional to the posterior error probability. So it's super confident in the PDAC cells. You can see this in the MS2 fragments, but like almost not at all confident in the U937 cells because it is so lowly abundant. In fact, you only identify one fragment in one of the scans, though it is apexing synchronously. So just because it's not confident doesn't mean that you can't extract valuable signal. In fact, we can, and we can get this identification and extract that signal. And this is, an identification that if you were just running U937 cells by itself, it's something that you would probably not identify. So these are some of the advantages that you would get with running multiplexDIA. We also did PCA analysis, and we clustered, we wanted to see whether our cells clustered together as expected. And so what we find is that, yes, indeed, single cells cluster well with the bulk, which I'm showing here with these squares, and they cluster well between the Q Exactive, the cells that were run on the Q Exactive, and also the cells that were run on the timsTOF SCP, suggesting that there are no biases in that sort of realm. So some of the conclusions that we can make from this is that with plexDIA, it's sensitive to a wide range of inputs from single cell analysis, which is what I just shown. And then it's also sensitive to bulk analysis, where you would run thousands of cells. We can get n-fold throughput by running these samples in parallel. Really high data completeness, again, because we're running these samples in parallel, and we can extract signal where we expect it to be. And we have accurate quantitation, which we benchmarked in several different ways. So with that, I would like to thank all the members of the Slavov lab, especially Professor Slavov, for all his mentoring and his advising, Andrew Leduc for preparing these single cells, Professor Demichev for all his work that he's done with DIA-NN, and Professor Marcus Rausler, and then Dr. Matthew Willits for running the samples, the single cell sets on the timsTOF SCP. I'd also like to thank our funders, and I would like to say that we have all this data that I've been showing is available on our website, where you can find links to the raw data, the Diane search pipelines, some of the process data, basically all of the process data that we use for the benchmarking, the cell cycle analysis, the single cell analysis, as well as the code that we used for the analysis, and as well as video tutorials and things like this. So thank you all for watching. I'm happy to take some questions if there are any.
Thank you, Jason. This was super insightful and really impressive work. I think I have to congratulate you and your colleagues. It's been really, really interesting to listen to this. I've seen the plexDIA resources, and they very in-depth, so definitely suggest that everyone have a look at those. We have some questions, so let's run through these. The first one from Julia. Let's see. Two questions on identification confidence. Question one, is the identification confidence affected by the increased complexity of the MS2 spectrum? Let's tackle this one first.
Yeah. So is the identification confidence affected because of complexity? It could be. Yes, it could be. Because the way that Diane is going to identify things, to my understanding, is that it's going to take correlations of MS2 fragments to the elution curve of at the MS1 level, among other things. And so if there is some interferences as a result of complexity, you could potentially have lower confidence in identification. There's other things like side reactions that might, reduce your coverage that way because you're literally going to have less abundant precursors. But this is something that's kind of resulting from anything that you label.
Okay. Maybe a follow-on question from me would be, do you check for the labeling efficiency in any way, or do you just assume that it's
yeah, absolutely. Yeah, and thank you for asking that. I probably should have mentioned that. Yes, we absolutely check the labeling efficiency. So we, I have a protocol for the labeling. It's relatively straightforward, though. There are some things that can go wrong. So I can share that protocol. But yes, what we do is do the labeling, and then we'll do like a shotgun DDA method, and we'll just check really quickly, running like a variable mod search that, in fact, more than 99% of the precursors that are being identified confidently are labeled fully.
Similar like a Silac approach where you check the
absolutely.
Perfect. So the second question from Julia was, how does the ID transfer behave in case precursors are actually absent in one channel?
Yeah, so this can operate in one of two modes. If you have, reasonable belief that the proteins across samples should actually be there, this is more so a general use case, then, you don't really have to rely on any sort of channel Q value filtering. You'll likely integrate 0 values. Sometimes you will integrate spurious signal. Usually that signal that you do integrate is going to be locally abundant relative to whatever channel is actually the more confident one.
Perfect, thank you. We have a question from Jan. First of all, it says great work. Are there any ideas on how to increase the n-plex, and is there a theoretical limit? So do you increase the complexity too much and generate too much interferences potentially?
Yeah, so thank you for that question. There's nothing fundamentally limiting going to higher plex. The reason we use 3-plex is because that is what is commercially available right now. But I think in the future with bulk, you could probably even go up to I think you could go up to 6-plex or potentially even higher maybe. But especially for single cells, because you're not going to have as deep of coverage, you can easily, I think, in my opinion, go to 10-plex or more.
Okay. We have a question from Isabel, also says, great work. How much multiplexing do you expect to be possible for DIA before spectrum complexity becomes too big of a problem? So I suppose it's related to the question from Jan.
Yeah. Mm-hmm. Yeah, and it depends on how deep of coverage you actually want to get, because as you do start to quantify, more things, your really complex spectra becomes more of a disadvantage. And so with really, large bulk where you want high, high coverage, I think that you could probably do a 5-plex or 6-plex, maybe push 10-plex, I don't know, but there are going to be limits at some point, sure.
Okay.
And this is, and I want to say again that this is less of an issue for single cell analysis because you're not going to get super deep coverage with
that. Wei has a comment, excellent talk, and he says the current version of plexDIA only enables the comparison of 3 samples. It also couple TMT Pro with plexDIA to further increase the throughput. So here he's asking that we can couple.
Yeah. So okay. So TMT Pro. I'm not sure if they're talking about the isobaric version or if they're talking about there's actually isotopologous, like super heavy TMT Pro that is available. To answer both those questions, if you do isobaric TMT Pro, you're not really going to be able to quantify any of the precursors that you get. The reason being is that you're quantifying the window that you get when you analyze precursors in parallel with isobaric mass tags. You don't actually quantify each distinct precursor. So it's not really amenable to doing isobaric analyses. But if you're discussing using isotopologous TMT Pro, like super heavy, TMT Pro super heavy, you could potentially multiplex that with MTRAC, but the thing is, they're not going to elute at the same time as something like MTRAC. So TMT Pro and MTRAC have different chemistries. And even if you combine them to say, maybe you form a 6-plex with that, they're not going to elute at the same time because of these different chemical labels.
Okay.
Hopefully that answers the
question. Did you see any unavailability shifts when you did the data acquisition with the timesTOF Pro? Or because of the labeling potentially, or
any shifts in the data acquisition?
Yeah. With the temps, you have to shift maybe the windows because you have now
you're right. For the timsTOF, yeah, for the timsTOF acquisition, we only use 5 different MS2 windows, which came out to be 3 DIA passive scans. And yeah, we didn't really go to super high M/Z ranges. We just kind of kept it where the precursors were concentrated. So I think we went from 300 M/Z to 800 M/Z.
Okay. You didn't have to shift it up. I think for your label-free DIA method, you had a slightly lower mass range, right?
Right. Yeah. So for the bulk analysis that we did, we used 480. We started at 480 and then went to, I think, 1480 or something like this. And then if I were to actually redo that, I would probably, maybe go down a little bit more. But, they're both fine.
Okay. Then we have another question. Can you comment on data points per peak? What optimization do you have to do on the instrument to get enough data points per peak?
What optimization? Right. So we ran it with 2 different data acquisition schemes. And I will say that the duty cycle that we ran on the bulk level was quite long. So for that, Diana actually does a really good job of identifying spectra, even if you have very few scans so that you actually identify it. And sort of miraculously, it does this. So for that, we kind of optimize the coverage by having fewer data points per peak, just for the bulk. But yes, your point for the single cell analysis, we did have to kind of optimize the data points per peak because the elution profile is going to be a little bit more shallow because the intensities that you're dealing with are so low. It's the peak is going to be a little bit shorter. And so, yeah, we had to shorten the duty cycle for that reason. Absolutely. And there's somebody in our lab, Georg Waldman, who is, making software that you can sort of plug and play and look at your DIA data and then optimize based on some reports that he's using the software to generate. So look forward to that.
Right. So I saw that you have the 2 acquisition schemes. You have the MS1 optimized versus MS2 optimized. So the MS1 optimized got more precursor ion scans versus more product ion scans for the MS2 optimized. What is your feeling in terms of which one should go for? What's the pros and cons of each approach?
Yeah, so what we found is that the MS1 and MS2 quantitation is actually both similarly pretty accurate. If I were to, do the MS2 quantitation, I would probably shorten the duty cycle a little bit, just so that you can have more data points per peak and not miss the apex, because that is important for quantitative accuracy. And that is the reason why we did so many MS1 scans for the method, for the MS1 optimized method. Yeah, so which one I would recommend is kind of up to you. I think that they both would work quite well.
Okay, awesome. I don't see any more questions. Here we go. There's one more coming. Given a Jerkett and new 937's different cell lines, I would not expect there to be 97% of the same peptides. How did you validate the plex data completeness is not due to spurious matches and false/false positives?
Yeah, and then this kind of has to do with looking at the quantitation that you would get with label-free DIA and plexDIA. And so matching up that quantitation, showing that there is good agreement between both these methods. But I suppose you're talking about the outer sect. So usually if you are integrating signal, the signal that you're going to integrate, and say that you extract signal that doesn't actually exist, most of the time you're going to get a 0 value. If you don't get a 0 value, usually the intensity is going to be so low that it might as well be a 0 value. But I think most of the proteome that you identify in a U937 cell and in a Jerkett cell line is going to be shared. And but like you say, there are different cell lines that are going to express different proteins. The question really kind of comes down to what you're going to do in the downstream analysis. If you're just going to impute the values anyway, then you might as well extract the signal if there is any.
Okay. I suppose so in doing the plexDIA is similar to essentially spiking with a heavy peptide. So you have the ability to know exactly when a particular signal is coming out. But so not only that, but also the peak shape of the actual signal should be identical. Have you looked into whether you can use the peak shape as another scoring parameter that you can enhance your ability to pick up signal?
Yeah, you're talking about the peak shape of the elution profile?
Yeah.
Yeah, absolutely. You're absolutely right. And this is actually something that Diane does. It takes correlations of the MS1 profiles to the most confident one, and it'll assign, scores on its confidence based on how well it correlates to this best channel, and does that at MS1 level and at the MS2 level. So yeah, this is actually something that Diane does for plexDIA data.
Okay. Awesome. So I don't see any more questions. So I'm gonna bring this one to an end. I want to thank you again for taking the time to present here on this platform. It was really awesome to hear about your work, and also thank for everyone for coming on and listening. And we will have this available as a recording in the next couple of days. So look out for the YouTube channel. And yeah, have a nice day, everyone. We'll chat soon.
Thank you, Soren.
Cheers, Jason.
Thank you, everybody.
Cheers, everyone. Bye.
Bye-bye.